Clinical evaluations of sleep deprivation typically focus on cognitive fatigue, brain fog, and localized neurological impairment. However, contemporary neuroscientific and epidemiological data reveals a far more insidious consequence. Missing sleep actively instigates a propagating, self-reinforcing cycle of social separation and withdrawal. This chart compares the absolute risk of premature mortality across major public health threats. Prolonged social isolation and loneliness increase the risk of premature mortality by more than 45%. This outcome is effectively double the mortality risk associated with severe clinical obesity, and it equals or exceeds the physical toll of smoking 15 cigarettes a day. This isolation is a specific neurobiological state called the loneliness phenotype, and it is actively generated by a lack of restorative sleep. Sleep serves as the obligate biological prerequisite for maintaining the social architecture of the human species, functioning as a requirement for survival rather than simple physiological downtime. To understand why a lack of sleep is lethal, we have to trace the exact physiological cascade it triggers. The damage begins with localized structural rewiring inside the brain, translates into behavioral withdrawal, and ultimately terminates in systemic cellular senescence. A lack of sleep initiates a biological weathering cascade that physically decouples an individual's chronological age from the rapid aging of their cells. Sleep loss immediately alters how the human brain processes social stimuli. This 3D functional MRI model isolates the theory of mind network, the medial prefrontal cortex, and the temporal parietal junction. This circuit is responsible for computing the mental states, emotional needs, and intentions of other humans. Following a night of restricted sleep, these core nodes suffer a massive reduction in task-evoked activity. By deactivating these specific cortical regions, the brain loses the cognitive prerequisite for empathy, destroying the capacity to accurately infer the needs or perspectives of others. This graph demonstrates the direct behavioral consequence of that neural impairment. Researchers tracked night-to-night sleep efficiency, the proportion of time spent asleep while in bed, alongside standardized helping questionnaires and real-world charitable donations. The data shows a steep, dose-dependent withdrawal of altruistic helping behavior. While the regions responsible for empathy shut down, a secondary system becomes pathologically hyperactive. This is the near space network, working in tandem with the amygdala. This circuit is dedicated to detecting interpersonal spatial threats and regulating the physical distance between bodies. Under sleep deprivation, this network exhibits an abnormal repulsion response. When subjects view approaching human figures, their brain demands a severe expansion of peripersonal space, forcing the individual to halt approaching peers up to 60% further away compared to their sleep-rested baseline. Simultaneously, sleep loss degrades the viscerosensory cortex, including the anterior insula and dorsal anterior cingulate cortex. This is the brain region that integrates autonomic bodily arousal with facial recognition to evaluate complex social cues. Without adequate sleep, this region completely loses its ability to reliably differentiate between threatening and affiliative pro-social facial expressions. Because the brain can no longer decode safe or kind signals, it defaults to a biased overestimation of hostility. This leads to profound deficits in interpersonal trust, especially in ambiguous social situations. The physical brain is effectively trapped in a continuous state of perceived hostility, having lost the exact neural hardware required to initiate and maintain human bonds. The architecture of this isolation is not limited to the internal mechanics of the sleep-deprived brain. It is heavily compounded by external perception, creating a phenomenon known as the social turnoff. This visualization tracks how a rested community responds to an exhausted individual. Independent observers, completely blinded to the sleep status of subjects in a study, consistently rated sleep-deprived individuals as unapproachable and socially unattractive. The external community unconsciously recognizes the asocial profile and actively shuns them. This initiates a rigid, bidirectional feedback loop. The neural rewiring forces the sleep-deprived individual to withdraw, while the rested community simultaneously enforces that isolation. The condition then spreads to the healthy population through the human mirror neuron system. These are specialized cells that fire both when we act and when we observe in action, allowing us to empathetically resonate with another's state. When a healthy individual observes a sleep-deprived person, their mirror neurons replicate the affect of isolation, causing the observer to report feeling significantly lonelier themselves. Escaping this state is further prevented by neurochemical lock-in. Sleep deprivation disrupts the precise pathways in the hippocampus that release oxytocin, the primary neuropeptide for social bonding. Without oxytocin release in the prelimbic cortex, the individual physically cannot encode new social bonds or retrieve the comforting memory of established relationships. A single sleep-deprived individual becomes a biological vector. Through behavioral withdrawal, societal shunning, biological homeostasis, the brain interprets isolation as an acute evolutionary predation risk, signaling an environment devoid of the safety inherent in cooperative group living. This flowchart traces the resulting endocrinological crisis. The perceived existential threat triggers chronic unconscious hypervigilance, sustaining the activation of the hypothalamic pituitary adrenal axis. The HPA axis is the body's central stress response system, and its sustained activation floods the body with cortisol, disrupting blood pressure control and autonomic function. Remaining in this continuous fight-or-flight state creates an immense allostatic load, a cumulative, toxic wear and tear on the body's organs. The relentless metabolic demand of hypervigilance eventually dysregulates mitochondrial energy production, leading to severe cellular exhaustion, particularly in the energy-hungry tissues of the brain. Anticipating physical injury or pathogenic infection from being separated from the protective group, the body executes a preemptive immunological shift. It abandons its basal viral defenses and actively drives the system toward chronic low-grade inflammation. This split-screen visualization compares the profile of circulating monocytes, the cells that act as the body's first line of immune defense. Even a single night of sleep deprivation radically alters the gene expression of these cells. The monocyte profile of a healthy, sleep-deprived individual shifts so dramatically that it matches the dysfunctional, pro-inflammatory profile typically observed in individuals suffering from severe clinical obesity. The evolutionary mechanisms designed to protect a stranded human backfire. The brain translates the emotional pain of loneliness into a systemic inflammatory cascade that damages the vascular endothelium, a response designed to survive a short-term crisis but fatal over long periods. The combination of HPA axis dysregulation and chronic inflammation moves the damage from the organ systems down to the microscopic level, weathering the human genome. This animated model shows the rapid accumulation of DNA methylation on a genome, quantified by an epigenetic clock known as GrimAge. GrimAge evaluates these chemical tags to calculate the true biological age of our cells. Under the sustained stress of the loneliness phenotype, this biological dial spins forward rapidly. This creates epigenetic acceleration. The cellular structures degrade so quickly that an individual's biological age physically decouples from and exceeds the actual number of years they have been alive. Simultaneously, chronic short sleep drives genomic instability by accelerating telomere attrition. Telomeres are the protective nucleoprotein structures at the terminal ends of chromosomes. The oxidative stress induced by the isolation phenotype causes these protective caps to erode significantly faster with each cellular division, destroying the cell's repair machinery. Causal mediation analyses identify this epigenetic degradation as the central biological mechanism linking social isolation directly to terminal cognitive decline and pre-dementia syndromes. Years of poor sleep leave a lasting biological toll, irreversibly accelerating the aging machinery inside the human cell. The loneliness phenotype represents a complete multi-system failure. It originates with the deactivation of the social cognition network in the brain, scales outward into societal shunning via mirror neurons, and cascades downward into an endocrinological crisis that alters DNA methylation. This diagram illustrates the ultimate danger of this pathology, the terminal bidirectional loop. Isolation and hyperarousal from sleep loss create unconscious vigilance. Interpreting this lack of connection as a threat, the brain is physically prevented from relaxing into deep restorative slow-wave sleep. Blocking deep sleep prevents physiological repair, stopping the glymphatic clearance of toxic proteins, and halting immune regulation. The individual is locked into a pathogenic cycle. Social isolation induces sleep disturbance, and the resulting poor sleep ensures continued neural damage and further loneliness. The evidence demonstrates that sleep and social connection are inextricably linked requirements for cellular integrity and disease prevention. Protecting sleep architecture is the mandatory first intervention. It is the only way to dismantle the loneliness phenotype, protect cellular integrity, and ensure human longevity.